there. All right. Welcome to Science Bubble. We'll just take a minute to wait for folks to log in. Thanks for joining. It's Popping the Science Bubble with Berkeley Public Library. What's new at the library? We have evening hours now. We're really excited about this. We're open on Mondays and Tuesdays at the Central Branch till 8 p.m. And then at the branches, Claremont, North, South, and West, we're open Tuesday, Wednesday, and Thursday evenings until 8 p.m. So this is something we just started in April, and we're glad to be able to provide more hours. Okay, so I'm just gonna go ahead and just introduce tonight's program. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they just find really interesting in general. The organizers are 4 graduate students at UC Berkeley: Madison Lemur, Oksana Andrick, Kaylin Zwang, and Daniel Bretthauer. This program happens every 3rd Tuesday of the month at 5:30 p.m. Starting next month, it will be returning to the library on site, but it will also still be on Zoom. And it will be happening in the first floor commons. This is the space that happened in pre-pandemic. And it will be at the same time, 5:30 p.m., 2nd or 3rd Tuesday of the month. If you're interested in checking out their past seminars, you can visit Popping the Science Bubble's website, and you can check out their YouTube page to watch a recording of this seminar or other ones they've had in the past. Also, consider signing up for the listserv to keep in the loop for upcoming seminars, and visit their website where they can post just a little more about each speaker. All right, thank you.
Thank you, Kelsey, and thank you all for being here. We're excited for tonight's speaker. As Kelsey said, we host a talk every 3rd Tuesday of the month and have researchers from a wide variety of departments on campus to share their interesting research with everyone. So if you're interested in learning about our future seminars, please join our listserv or the link that is on the screen right now. And you can also check out our website, Twitter, or YouTube. For tonight, we are excited to have Naren Kumar and Ruwan Thilakaratne with us to present. And this is an open format talk, so we encourage questions throughout the presentation. You can submit your questions either through the chat function or the Q&A section, and we will ask it in real time. And we will also have time after each talk for more questions as well. So to start us off, Daniel is going to introduce our first speaker of the night.
Yeah, our first speaker is Naren Kumar. And so Naren grew up interested in understanding how tiny creatures called microbes in the soil made life possible on Earth. And so today, his research brings the same curiosity to studying bacteria that can cause serious infections in humans. And as a postdoctoral researcher at UC Berkeley in the Physig lab, he tries to understand why the bacteria become resistant to antibiotics. And so in his downtime, he likes to communicate science through his podcast, pH Defined, and make the profession of science more accessible to everyone. Take it away, Naren.
Thank you so much. First of all, I'm quite excited for the format that we have over here, and I'm going to jump right in. So today I'm going to be talking about something that's really close to my heart, and I think it's going to be a major issue, something that we're tackling even now over the years. So I'm going to be talking about antibiotic resistance, um, and then focusing a little on some of the research we're doing in the lab. For instance, the Fleisig's lab at UC Berkeley, which looks at intracellular bacteria and Pseudomonas aeruginosa, which is a bacterial pathogen. So just a quick disclaimer here. All the thoughts that we're going to present today are more summaries that can help provide a better understanding of the topic. Sort of a more in detail discussion can follow if there's anything that's related. So I'd like to start by asking the question, and this is something that blew my mind when I started thinking about how these areas were interconnected. And what do we think really connects humans, fungi, the practice of surgery in the medical field, plants and animals? And if you think about it closely, it's other than the fact that we are interdependent and we require each of these things to survive, they all involve the use of antibiotics at some point in its life cycle. If we get an infection, humans would be given an antibiotic. Fungi are the majority of fungi produce certain chemical compounds that can be used as antibiotics. In surgery, certainly we need antibiotics. In agriculture and animal husbandry, we use antibiotics. So if you think of it, antibiotics is not just a biomedical or a human problem, or used, but it's more widely used across society in the present day. Here I would like to bring in a short conundrum that sort of occurred to me when I was a high school student. Was every time you would get the cold, you'd be prescribed antibiotics along with the cold. And this is really confusing. And the reason for that is antibiotics are drugs that target specific processes in bacteria. A cold that may be viral in nature is not, antibiotics are not really treating the viral infection, but they're only treating the secondary infection that you may get. And this was quite a trend when I was growing up in India, because early in the 90s, if you went to the doctor for a flu, you would be prescribed a course of antibiotics for 5 days. Now, with current increases in resistance, as you may have heard, this practice has sort of reduced, where now we have changed to the process of having the flu and then going past it without using antibiotics, but using palliative care or using some kind of medication that subsides symptoms. But this is something that's quite important to distinguish, is when we say antibiotics, we're really talking about drugs that kill bacteria, but not viruses. Are antibiotics new is really the question. And if you look back far enough, antibiotics have been around for many, many, many centuries. Not in the form that we currently use them, but they have been there for many centuries. And here is an example of, so this was a paper that was published in 2015, which actually took the recipe that was mentioned in the Ball's Leak book, which is a 10th century text, Anglo-Saxon text. They made it exactly the way they showed it in the text. And it was the perfect treatment for a stye infections that you get in the eye. And one of the things that people say is, when you look at ancient texts, it's always complex. If you look over here, you require equal amounts of an alum species, garlic, you need to pound it together, ox gall. You need to put it in a brass vessel. They found that you don't need any other kinds of vessels. You absolutely need a brass vessel. And then you need to let it stand for 9 nights and before it was actually active. So That's kind of a bone that modern day medicine picks with ancient medicine, is that how do you know what's really causing, improving your symptoms, um, and making you feel better. Later, you can see in 15th century text where they had different kinds of mold growing on bread, and they would take a certain kind of mold and apply it to a certain lesion, and you'd be able to cure tropical infections. But it was not until 1928, and this is quite a famous story, where Alexander Fleming was attributed with purifying penicillin, discovering penicillin as they call it, but it was really the efforts to try to purify it after the discovery and then which leading it to mass production, which ended up getting the Nobel Prize shared with two other people. And we really call this the golden age of antibiotic discovery, because this is where across the world, people were discovering novel compounds that were produced by nature, fungi, or other species, and then use them to treat bacterial infections and make the drugs that we know today. Um, so the question now is, we have antibiotics, but how do they work? And in terms of bacteria, so bacteria, unlike viruses, are living. They have their own replicated machinery. They have a structure. So here we're looking at a bacterial cell. You have an outer membrane, an inner membrane, and you have all this nucleic acid or a single chromosome. Um, and you have all these different components. Now, I'm going to go into a slight more detail, but antibiotics inhibit specifically protein synthesis, DNA, cell wall, and each class of antibiotics may be working in a different way. And the way to think about it is that the drug and the target that it's going after is more of a lock and key mechanism, where there needs to be a perfect fit, otherwise it will not work. And the way bacteria find a way around drugs that we can use to target them is that they can change this lock and key in many ways so that the drugs don't work anymore. One of the ways is they would destroy it or cleave it so that you don't really have the drug anymore. Another way is it would modify it so that it's not active anymore. In another form is that it actually uses something totally different. It's like changing the lock at your home. If somebody found your key, you would get a different lock and you put it on, and nobody can get access to your home anymore. You can alter the target. You can change the address. You can say, this is not where the drug binds, but it goes somewhere else. Bacteria can stop the uptake altogether so that very few cells actually get the drug that we're giving, or they may take it but then push it out rapidly so that the bacteria don't really get affected by it. These are all the many ways that we know bacteria can change antibiotics that's leading to resistance. But the fact that we're interested in is, and also there's the problem of multi-drug resistance, which I won't go too much into today, but it's when bacteria become resistant to more than one antibiotics. The particular problem that we're interested in understanding in my research is, what about bacteria that go inside cells? Now, bacteria we typically think of as swimming around the cell, causing an infection, or being in the tissue when we get a skin lesion. But what happens if these bacteria were to go inside your own cells, and then what are the challenges associated with it, and how do you target it? So here I'm listing all the classes of antibiotics, and here I'm showing whether each one of them actually go inside this eukaryotic cell, which are our host cells. And you can see that a lot of the antibiotics that you may be prescribed target these bacteria that are outside the cell, but very few other antibiotics actually go inside the cell in any appreciable amount and then target this population. So that's what we're interested in understanding. We're taking, I like bacteria, and it makes more sense to me. So I take a bacteria first approach to understanding this. And this is a movie that was sort of captured early in '97. The timestamps are real, and this was taken by Dr. fly's egg. And what this shows is that shows the same thing. So this is a bacteria killing the cell from the outside. As you can see that the bacteria sort of makes its way through the entire cell layer. But over here, you can see that the bacteria are outside the cell, but slowly you can see some bacteria make their way inside the cell. And then it's not as toxic as this particular bacteria, but you can see this intracellular replication happening. And the question that we've been interested in answering and the lab has been trying to answer over the decades is, how do these bacteria replicate inside the cell? What are the factors associated with it? How are we doing on time?
I think there is still about 7-8 minutes.
Okay, good. Yeah, so some of the questions that we're interested in answering now is like, now you can see these bacteria replicating and filling the cell, and the cells look relatively healthy, unlike what we have over here. So the question we're trying to ask is, what are the bacteria doing inside the cell? And what happens to these bacteria? What about their sensitivity to antibiotics? If this were the case in an infection that we may get. So here you can nicely see them fill the cell. So some of the tools that have been developed now to use this is we use some amount of genetics. I'm going to spend some time because this is important and sort of the core tool that we use to understanding bacteria inside cells. So we've all heard about genes. Genes are stretches of nucleic acid that make proteins that go on to fill the cell. But what we don't talk much about is that the gene actually has a lead up sequence, which is called the promoter sequence, and that's called the anatomy of the gene. So the promoter tells when this gene needs to be turned on, and the gene itself tells us what needs to be made. And we can sort of leverage this in some form, is that we can now take fluorescent proteins that are made by other organisms. They're safe. So you can get them in different colors. They have green fluorescence, red fluorescence, and yellow fluorescence. And now you can combine the promoter region with the green region. So when the gene turns on, or when it signals to turn on, you're making a green protein instead of the gene. And that's a little complicated, so I'm going to spend a little time. So in this case, where I'm talking about the anatomy of the gene, you're making the protein that is associated with it. But in this case, you're only tracking when a protein turns on, which tells us we can look at gene expression at the molecular level, we can look at the dynamics of it, and it's modular. So you can take any promoter region from a certain gene and match it with your gene of fluorescence of choice, and then you can track it over time. So one way and we use it is to study this intracellular bacteria. So here I'm showing you cells. So these are epithelial cells from the cornea. This is cell culture, so we maintain them in a dish. And what you would see is you can see the nuclei over here in blue, and we do live imaging on it over 24 hours. So every frame is captured 1 hour after, at 1 hour intervals. So you see the nuclei, you see the cytoplasm, and in green, you're going to see the bacteria. And so here is an example of a single gene, and here is an example of another gene. And what you can see is when the bacteria that express the gene get inside the cell, fill the cytoplasm, as I showed you earlier, and then replicate rapidly. And over time, you see the cell die and hit as soon as it turns red. But when you say, okay, we take the same setup, now I'm not changing anything except the promoter region, which is telling which gene is being turned on and when it is being turned on. And here you can see on the right that you can see that when only the bacteria that express this gene are confined to a smaller space inside the cell. which suggests to us that there are two populations inside the cell, one that is filling the cytoplasm and another one that is more confined in a small space. Okay. So now using this tool, we had a question of like, now, what does this mean for antibiotics, and can we sort of close the loop from where we started? So now considering antibiotics that are going inside a cell, they go inside with a process of diffusion. So when you add an antibiotic, it's highest on the outside, and then it may be lower on the inside. This was also published in a recent publication that we shared. And we say, okay, when bacteria express this one gene, how sensitive are they to this antibiotic? And it turns out that you need at least 4 times as much antibiotic to debilitate the bacteria that are inside. So you can see this by the green fluorescence here, but very little fluorescence over here, which suggests that the bacteria have now died. And you also see that the cell are healthy. So it takes about 4 times as much of the antibiotic to debilitate these bacteria that express this gene, but the same amount of antibiotic is not sufficient to debilitate the bacteria that express this particular gene inside the cell. Which suggests now we can study these two populations and then ask the question of why this particular population becomes resistant, what are the factors that may be involved in it, and how do we go on to understand this better. So in summary, I think what I hope the big takeaway messages are that antibiotics are drugs that target bacteria, and resistance is a growing problem. And resistance, we need to come up with novel ways to sort of tackle resistance, and that's sort of my passion. I'd like to understand this more for other bacterial species. We need to understand what happens to treatment when bacteria can get inside cells, as they may be difficult to treat. And while an understanding of the antibiotics themselves are that these were discovered from natural sources, but have since been modified to work better on infections. And we need to keep pushing along that journey to sort of make this better, but also take this holistic approach that I mentioned earlier, as not seeing antibiotics as a health problem, but seeing it as a community problem where you may have challenges on livestock, you may have challenges on agriculture that may need to be addressed, and the problem may be bigger than just human health and diseases. And we need a newer class of antibiotics, and we need to tackle the problem of multi-drug resistance, because in the US alone, that's a huge problem, and globally, that's a problem that people have been calling a silent pandemic. So that needs our attention. With that, I'll take any questions, and thank you. Thank the organizers for the invite to share our work and present at this meeting. I think it's a great platform. I'd like to thank all our lab members, our PI, Dr. Fleisig, Dr. Evans, and all the other lab members. So thank you.
Yes, thank you very much for that great presentation. So please put any questions you have either in the chat or in the Q&A section, and I'll give that a few seconds. Otherwise, I'll use my host responsibility, my host privileges to be able to ask the first question. Alrighty, so while people are probably typing, then I'll go ahead and ask the first question. So I was wondering, towards the beginning of the presentation, you had mentioned that there are a lot of different ways that some bacteria could potentially be more resistant to these antibiotics. And so I was wondering, are some of these particular ways of being resistant more common than others? And kind of, can we use that to help more target the antibiotics to get rid of the bacteria?
Yeah, that's a great question. I think I hope the answer was as simple, but I think the mechanism that bacteria use to prevent killing is specific for the class of an antibiotic, because each antibiotic targets a certain, say, target inside the cell. So one may go to the ribosome, so that may have a particular mechanism of changing the susceptibility. So yeah, it would be different for each class of antibiotics, and it's not really predictable in ways. There are definitely prevalence. So you can say that a certain mutation is associated with, say, gentamicin resistance. But it's harder to say if the same mutation will cause resistance in a different organism or a different bacteria. So it may be bacteria specific. And a lot of work is being done now and by others in the field, where they're trying to understand how different mutations combine together to give a certain effect. Because if mutations don't work predictably always, and you may have more than one as well. So yeah.
Thank you very much. Look like there is a question in chat. If bacteria are more resistant to antibiotics while inside human cells, is it believed to be just an issue of antibiotic concentration, or can human cells help the bacteria by metabolizing these compounds?
So the first part of the question, that's a great question. If you think, it is definitely a availability issue, where when you think of antibiotics getting inside human cells. And the thing that I did not talk about here is when we administer the drug to humans, the amount of antibiotic, let's say 50 milligrams per mil is what the capsule says. The dose in circulation is not 50 milligrams. And what you need in circulation is you need at least 10 times higher in your blood than what the bacteria can tolerate. So so at least the first part of the question is an availability issue that needs to be addressed. And if we can get more inside the cell, it may be better for to target these bacteria. The second part of the question, could you please clarify that? Can human cells help the bacteria by metabolizing these compounds? Is this referring to the antibiotics itself?
Yes.
Okay. Yeah. So that could be one way. It could be one way where you could sort of have a form of the drug that gets better inside the cells, and then human cells change it. That way you have a higher concentration of the active form inside the cell, closer to the bacteria. I'm not aware of a drug that works that way for intracellular bacteria, but there may be somebody working on it, and I just don't know about it. But yeah, it's possible.
Oh, sorry. The question was asking more, can human cells, or I guess whatever organism cells, actually break down the antibiotic so that it, like, even the amount that gets into the cell further shields the antibiotic or the bacteria from the antibiotic?
Got it. Sorry. So yeah, I mean, that's totally possible. Again, that's a thing we're trying to understand now, is like, are the bacteria doing it, and what is it? Is it breaking down of the product, or is it removing the product, or is it sort of changing its own cell to be more resistant to the product? So at the moment, we're thinking of it more as tolerance rather than resistance. It's like they're surviving, but we don't know whether they will continue to survive long term.
All right. Are there any more questions?
Okay. I think if anyone thinks of any last minute questions, Naren should still be around, I believe, for the next few minutes. So you're free to put them in the chat where he can answer them. Or of course, we always take questions after talks as well. if there's any you think of later. But for now, I will introduce our next speaker, Ruwan. He's a doctoral candidate in the Epidemiology department at UC Berkeley School of Public Health, where he's advised by Dr. John Balmes. He studies how we can make our environment safer, particularly for children, currently focusing on early life exposure to metals and their health consequences. Ruwan is an East Bay Area native who grew up in El Sobrante and the son of immigrant parents from Sri Lanka. Outside of research, he enjoys reading classic literature, running, and meditating, and we're very excited to hear his talk today. So please take it away.
Thank you, Madison. So hi, everyone. It's nice to be here, and I want to thank the Popping the Science Bubble team for allowing me to give this talk and share some of what I work on and what I love doing. Naren's talk was also super interesting. Thank you, Naren. It's a hard act to follow with all the videos and really great graphics. So I hope I'm not kind of providing too much information here, but I'm hoping this will be informative for folks about how we think about artificial food dyes and their health effects. So I will go ahead and share my slides and get started here. All right, let me just a little more setup here. Okay, and I'll go full screen. All right, so yeah, my name is Ruwan. You already heard a little bit about me, and I'm going to be talking today about my work. This actually is not what I'm currently working on as a doctoral student, but I worked on very recently, and I thought was worth sharing with the public because it's an issue of great interest to many, and especially to parents, which is the potential health effects of artificial food dyes. So the title of this talk is Shades of Risk: How Safe Are Artificial Food Dyes for Children? And before I proceed, I do want to make a disclaimer. The views that I'm expressing here and other materials for this talk are my own and don't necessarily represent the views of the Office of Environmental Health Hazard Assessment, or OEHA for short, which is an acronym you'll hear a lot in this presentation. So the California Environmental Protection Agency, or CAL-EPA, or the state of California. Obviously all these organizations were involved in this work, which is why I'm making this disclaimer. So just briefly, I want to just outline what I would like to talk about today with you all. So first, I just wanted to clearly define artificial food dyes and their uses, and then talk briefly about the history of artificial food dye policy and potential health concerns. And then get into a little bit of the basics of a process we in environmental health call risk assessment, and kind of what that means and how it works and how it's applied here. And then talk about a report that was published recently by OEHA on the potential health effects of artificial food dyes. And then I'll end with some concluding remarks. All right. So artificial food dyes, I'm going to call them actually artificial food colors, or AFCs, from here on out, have several, synonyms. So they're also known as food dyes, synthetic color additives, and there are other terms as well you'll hear. These are actually derived from petroleum, which not a lot of people know. And their specific usages include enhancing the visual appeal of foods, particularly for children. You can imagine, soft drinks and candy and ice cream and things like that. And also to mask natural variation in color to create more uniform color, and also to distinguish different medicines. And there are other purposes as well that are not listed here. And so for the AFCs approved by the US Food and Drug Administration, or FDA, which is the governmental body responsible for ensuring all of our food and cosmetics and drugs are safe, they approve of 9 total AFCs. So 7 of them are listed here: blue number 1, blue number 2, green number 3, red number 3, red number 40, yellow number 5, and yellow number 6. And you'll have likely seen these if you've looked at the ingredient labels of foods that use these dyes. And these may go by other common names like Allura Red and Sunset Yellow and others. There are two other dyes, Orange B and Citrus Red #2, that are both also approved for use in foods, but are used way, way less and for very specific purposes, and so are not going to be in the scope of this talk. And so I want to actually just immediately jump into kind of understanding what the US FDA recommends in terms of intake limits for artificial food dyes. And these kinds of limits also apply to other chemicals and compounds that we include in our food for various reasons. And so a key concept is this concept of an acceptable daily intake, or ADI. And this is basically the maximum level of daily intake of some chemical that is believed to not cause adverse health effects. So it basically answers this simple question of how much of this can I eat every day and still be safe? And the ADI is calculated actually as this quotient here, where we divide what's called the NOAEL, or NOAEL, which stands for the no observed adverse effect level, by a safety factor, which is usually just a number that's a power of 10, like 100 or 1,000. And the NOAEL is, I try to demonstrate the NOAEL here on the right, which is just an image of basically showing kind of how we understand what level of a chemical is safe. And so what happens is studies are done in animals, for example, often rodents like mice or rats, where they'll give increasing amounts of a chemical to the animal and see at what point do they see adverse effects in any way in the animal. And so the highest level of exposure that does not cause a health effect is termed as the NOAEL. And so that is how the NOAEL is defined. And so for the food dyes, or AFCs, the NOAELs are based on animal studies with that showed that there are various outcomes like effects on the gut, effects on lungs, and growth of juvenile rodents were impacted by exposure to the dyes. And these were done in the 1960s through the 1980s, so fairly old. And a major critique of these studies is that there's not a lot of consideration of neurobehavioral outcomes like attention and activity levels. And so the few studies that did look at those outcomes, were, because they were older, did not use the most sensitive or kind of, the best basically measures for measuring attention and activity and other kind of brain activity related outcomes. So I just want to set that as kind of the backdrop, and we'll get more into what more recent studies might be saying. And so first, I also just wanted to give a brief history of AFC science and policy in the US to kind of bring us to current day and what's being done now. So in the 1950s, that should say the 60s to the 80s, animal studies were done that established the ADIs. And then in the 1970s, a physician named Ben Feingold, who was an allergist, basically found in his clinic that when he removed artificial food dyes and some other types of compounds from the diets of children impacted by attention deficit problems, that their symptoms seemed to resolve. And so this kind of evolved into this what's called the Feingold diet that grew in popularity in the 70s. And this kind of diet and this kind of interest in artificial food dye effects on attention and activity morphed into a real scientific interest in this. And so there's studies throughout the 80s and 90s, toxicology studies and epidemiologic studies about neurobehavioral toxicity of these dyes. And in the 2000s, there were even several large randomized studies that found harmful effects on some children's levels of activity and attention due to attributable to exposure to the food dyes. And so in the early 2000s, several European countries, including the UK, made substantial policy moves to address this issue, and actually added either added warning labels or to foods that contained these dyes to warn parents and others that there may be adverse effects from them, or even outright ban certain synthetic food dyes. And so the U.S. A little bit later in 2011, was charged with the US FDA was charged with trying to review the evidence to understand whether the U.S. basically believed that these dyes could cause neurobehavioral effects. And actually, through their review, they ended up concluding that the evidence was insufficient. And so the issue was kind of laid to rest here in the U.S. But in 2021, OEHA, as I mentioned, is a California agency, was charged by the California legislature with conducting a risk assessment to understand the health effects of food dyes, using all of the available literature to date. And that was published in 2021, and an image of the cover of the reviews of the report is on the right there. It's available publicly as well. They concluded that there were adverse effects of the exposure to these food dyes on some children's levels of attention and activity. And so we're going to get into why they made that conclusion and what that means. So first, I wanted to take a very brief detour into just understanding what this concept of risk assessment is, because this is something that informs a lot of the regulations that we have that affect your life and everyone's lives who live in the U.S. And elsewhere, in terms of what they're exposed to and what governmental policies are designed to mitigate in order to keep the environment as safe as it can be. And so risk assessment basically has four major steps. The first step is on the left, there's hazard identification, which is basically, does this chemical cause health effects? Is there scientific reason to believe that? And then we have dose response assessment, which is conducted at this simultaneously with exposure assessment. And so dose response assessment asks, well, exactly at what level are there harms from exposure to, like, at what levels of exposure to this chemical do we get health problems? And then exposure assessment asks, how, in the public right now, to what extent are people exposed, and how much, and which groups, and those kinds of questions. And so combining those, the answers to these questions, we can get what's called risk characterization, which basically just tells us, okay, given that health effects occur at this level, and given this is how much people are exposed, we can expect that there's X level of health problems related to this exposure currently happening in the population. And so that then can drive efforts to reduce exposure through various policy means and other actions. And so that's basically what was done in the OEHA report. And so I want to go through just kind of several different evidence streams that allow us to like kind of answer these questions, right? So I mentioned there were studies with animals to establish the ADIs, the acceptable daily intakes, and there are also, of course, human studies and studies in cells even to see what happens to different types of cells and human tissue in the lab when they're exposed to different chemicals. And so it's really an effort, risk assessment is really an effort that combines all of our sources of knowledge about a chemical's health effects. And so I just wanted to briefly go over kind of the different parts of this. So the epidemiology of this, so that basically the study of the studies that have to do with actual humans getting exposed to the AFCs. There were several dozens of randomized studies that were included, including, as I mentioned before, 2 large randomized controlled trials that found significant adverse effects of the food dyes on levels of attention and activity levels in the kids. And these are kind of symptoms that we associate often with the well-known condition called attention deficit hyperactivity disorder, ADHD. And so the results of these studies, Inuiha's assessment could not be explained by bias, due to issues with the study design, et cetera. In other words, based on these human studies, there's good reason to think that there were real harmful effects of the food dyes on the children's behavior. And then the animal toxicology, there were a lot of studies done in rodents, including mice and rats. They included, more recent studies that found that exposure to any of several dyes, including red 3, red 40, yellow 5, and blue 1, to name some specific ones, affected activity levels and the brain structure in juvenile rodents. And multiple studies indicated that lower noles, so again, the nole is this level of the chemical at which the highest level of the chemical where we don't observe an effect. Anything above that, it should be causing an effect according to these experimental studies. These newer studies indicated that there were the lower noles than those currently used by the FDA, might need to be instituted. And the FDA studies, again, were not based on neurobehavioral outcomes. And overall, the evidence supports a short-term effect of individual and combined dye exposure on behavior. So they tested both individual dyes one at a time and also mixtures of the dyes, as as you would expect that to occur in real life, right? As people consume multiple foods that have different dyes in them, and even the same one food can have many different dyes. So then that kind of takes us to exposure assessment. And this is actually the part where I was most involved in this work. So I'll just describe the results in slightly more detail. And I'll be better at answering questions about this part of the work, but I'm happy to take any questions about any of it. And so for this, we used, basically the question was, what is the level of exposure in the US population to each of these dyes? And how does that relate to the ADIs? Are they above or below the ADIs? And so we use the National Health and Nutrition Examination Survey, or NHANES, which is a representative survey of the US that has for most participants 2 days of dietary recall information. So basically what a given person consumed in the previous 24 hours. And so 2 days of that information. And we were able to link that dietary information to concentrations of the different AFCs measured in a laboratory, and this was actually done previously to this work, but we were able to use the concentrations estimated by this other lab across many foods that were purchased in just at the various grocery stores. And through combining those sources of information, we can then estimate daily average exposure in units of milligrams of the AFC per kilogram body weight, because obviously people who are bigger are going to eat more and have more exposure. So we want to account for the kind of body weight per day. And so we're able to compare those to the ADI. And this is kind of the component that we would call risk characterization, where we're now determining, okay, are these levels of exposure concerning for health effects? And so I'm going to show a table here. I don't want to focus too much on any of the specifics. I know there are a lot of numbers, but just wanted to point out that the bolded numbers here are values greater than 1. And what the numbers are representing is the ratio, or the kind of basically the division of the level of exposure divided by the ADI. So ADI greater than 1 means that exposure was greater than the ADI. And so we had, we observed in this work for red number 3 specifically, this is for one of the dyes, that there were many cases where there were exceedances of the ADI. And remember, these are ADIs not even yet based on the neurobehavioral outcomes that seem to be more sensitive to the food dyes. So even with these older ADIs, based on these older studies done 50 years ago, we're seeing some exceedances for red number 3. We did not see any exceedances for any of the other dyes. And these are done specifically for children and women as well, and pregnant women.
Ruwan, we do
Yes.
have a question asking, is dye intake measured as like serving size units?
Oh yeah, that's a good question. So the dye intake is measured. So if I go back to so it's in units of milligram per kilogram body weight per day. And so what we do is basically take the concentration in the food. So let's say someone eats a piece of candy that has some number of milligrams or micrograms of the dye per gram of the food. And we then multiply that by how much food the person ate how much of that food the person ate. And then that gives us, basically how much in weight of the dye that the person had as intake. And so that's kind of how we measure how much dye was consumed. So it's basically the weight of the dye. And that's to get, to this, to make it comparable to the ADI, which is based on the weight of the dye per kilogram body weight per day. Does that answer the question, Naren?
He's saying yes. Great. Thank you.
Okay, great. Yeah, no problem. Feel free to ask any questions. So again, let me move forward. So what I was going to mention is with this next graphic, we're able to then look also at, what foods specifically are contributing the most to overall exposure in children, exposure to these dyes. And so the culprits you would expect are the ones that we found, like things like icing, ice cream cones, sprinkles, like baking decorations, like things that you would expect would contribute a lot because they're very colorful and very brightly colored. And this is just for red number 3. There were, the layout looks a little bit different for different dyes based on kind of which foods they tend to be used in. We also, just wanted to briefly mention that, we try to make it a priority to look at disparities in exposure by socio-demographic factors and socioeconomic status. And so we found that Black children and women had higher levels of exposure, significantly higher levels of exposure than children and women of other race ethnic groups. And we also saw higher levels of exposure in individuals who had lower income compared to individuals with higher income. So there's some kind of socio-demographic trends in exposure as well that may be driving inequities in health. And so just to summarize, then the exposure assessment findings were that for red number 3, we found multiple estimates that exceeded the ADI. We found that icings, ice cream cones, and fruit drinks were important sources. We found that lower socioeconomic status is correlated with higher exposure. This wasn't mentioned before, but as I mentioned towards the beginning of the presentation, actually, that medicines, also have these artificial dyes in them. We measured levels of the dyes in over-the-counter medications, and we found that levels were pretty low compared to foods. So that was good. And the highest levels of exposure came from young children consuming cough syrups. So cough syrups tended to have the highest food dye concentrations. So this is my last slide, and I just wanted to kind of summarize again OEHA's conclusion that, or I guess reiterate their conclusion that they've found that artificial food dyes may cause behavioral problems in some children. And so that scientific assessment is based on all of the kind of science that I've discussed so far. And so then the question is, of course, what do we do now with these results and the findings of this risk assessment? And so one is obviously that further research is needed. So we need to better understand exactly which children are affected, because it doesn't seem like all children are affected equally. More animal studies are needed with, again, high quality, basically endpoints or kind of outcomes to be measured for capturing neurobehavior. And there's also interest in what the long-term impacts of exposure might be, because we have these dyes on, in a long-term basis, and there may be long-term impacts, and that hasn't been studied yet. And for public health practice, obviously, as I mentioned, some countries have taken, pretty strong, positions to either ban or add, warning labels on foods that contain these AFCs. But here in the US, that has not been done yet. And so just at home, parents can choose, of course, to reduce children's AFC intake. There are now many products that have natural fruit and vegetable-based dyes in them that can serve as alternatives to the artificial food dyes. And so that's pretty much all I wanted to share today. I just wanted to highlight the acknowledgements. I just wanted to acknowledge many scientists who contributed to this report, and especially Rosemary Castorina and Asa Bradman there on the left, who Rosemary is still at UC Berkeley. Asa is now at UC Merced, but two of my really close mentors throughout this work, who are excellent exposure scientists, and other Berkeley contributors as well. And over at OEHA, some great mentors and scientists as well, Mark Miller and Melanie Marty, as well as some other OEHA contributors to the report. And I also want to thank, of course, Popping the Science Bubble again for the opportunity to present here today. And yeah, those are all my slides. So yeah, I'll take any questions at this point.
Great. Thank you so much for that talk. We'll wait for questions to roll in. Again, as audience members, you are free to type them through the Q&A and chat. But while you are typing, I might kick us off. Are these dyes, and I apologize if you said this earlier in the intro, are they generally only food-based, or like are these dyes that are put in a lot of a variety of things? I'm thinking of like Play-Doh, but just like a bunch of like colored stuff like that.
Yeah, it's a really good question. So this work focused mainly on these dyes because they're approved for use in foods by the FDA. But yeah, the kind of act, the federal, act in the law sense that regulates these dyes includes other things like drugs and cosmetics as well. I know cosmetics is a big category. I'm actually not sure, about like Play-Doh, though that, obviously that is very, very brightly colored. But I think, these dyes are pretty, they're pretty versatile and, serve a lot of different purposes in everything that we use and a lot of what we eat. So yeah, I wouldn't be surprised if they were used in Play-Doh and other things. So yeah, they're quite common. Hopefully people aren't eating Play-Doh as much as food though.
Maybe not, just thinking of the 3-year-olds that play with it.
Yeah, I'm sure some of it gets into their mouths for sure.
Okay, we do have a few questions coming in. I'll start us off with an audience question. Saying, hi, big fan. How do you go about informing policymakers to possibly reduce these dye exposures? By disseminating the data, how do you foresee implementing the data for more policy regulations? As you mentioned, other countries banning these dyes. Yeah,
and Thaven is actually one of my good friends, so I'm glad he could join. I shared the link to this chat with my social media network. So so thanks for coming, Thevin. He's not a planted audience member or question. This is a tough question, as you can tell, not an easy one. But yeah, it's a really good question. So I think so in terms of the next steps for policy, I think we as scientists, kind of the most we can do is really just provide the best, most accurate and true information that we can about a given subject. So this FDA commission in 2011, was charged, as I mentioned, with assessing the evidence for neurobehavioral effects, and they came to their conclusion based on the evidence that they had. And so I think, it really takes kind of the will of the public and will of legislators to do something about this kind of issue. There are, of course, many issues that policymakers face, and this is just one of them. So, you know, I really think it comes from, you know, the level of concern that we have about the health issue and the potential consequences of these exposures. So the ADIs, again, that are currently used are based on pretty old data, and there's good reason, you know, OEHA thinks, and I think also it's good reason that, you know, those ADIs should probably be lower based on newer studies. And so that might, you know, draw more attention, you know, if we do find, you know, if there are headlines like, you know, kids are exposed to, you know, weight levels that are way too high based on what we know about when these health effects should occur. So I think, yeah, it has to do with the science advancing and with there being an interest by other institutions to do something about this. And yeah, Europe in general is a, you know, definitely interesting case, and you know, definitely they follow a different philosophy. They're kind of, policymakers follow a different philosophy, often called the precautionary principle, which is just, you know, the idea that, you know, if something seems bad, or there's, you know, some evidence to say that something is bad, we should probably, you know, address it and before, you know, coming to a final conclusion about it scientifically, which can take years and decades, you know, to avoid potential harms. And yeah, we might have been wrong, but at least, you know, on average, we're going to be protecting people, and hopefully doing that more often than stifling, you know, the great, you know, uses that might come of different technologies that have, you know, different technologies and products that have different health risks. So, yeah, I think I foresee, you know, a stronger interest in this issue, especially in California. You know, California is a trailblazer in environmental, everything environmental, really, in the U.S. You know, this is a pretty landmark report, you know, which was, you know, definitely noticed by the FDA and, you know, federal agencies. So, you know, the hope is that, you know, this brings more attention to this issue, more science, good science to really understand it, and you know, better policy to protect kids, because I think this is something, you know, all sides of the political spectrum can definitely, you know, understand and get behind, is, you know, what can we do to protect our kids. So yeah, so thank you for that question, and yeah, it's a great question. So thanks, Devin.
Great. And I think that leads into our next question pretty well, which is asking, like, specifically, you know, after this study, what are the next steps, especially considering they kind of change how we think or how we understand these dyes? You know, what are the next steps for this study? And like, do the FDA then, you know, re-review their guidelines? How long does it take to get there, kind of thing?
Yeah, these are really good questions that I honestly don't have good answers to, because they're kind of, this is, I mean, I can tell you definitely where the science should go, and those are explicit recommendations from the OEHA scientists that were included in the report about, you know, having, you know, more, you know, better studies in animals of the neurobehavioral outcomes, and you know, understanding of potentially like differential genetic susceptibility to these adverse effects. So studying how like genes might, you know, different gene based on your genetics, how that might increase or decrease your risk of effects, as we see that is true for a lot of things in the environment and in medicines as well. So yeah, there's a lot of room for the science for sure, and that will take time. I think in terms of policy, I don't really know exactly what the next steps are. There was actually so this you know, kind of this investigation done by OEHA on this issue, and this risk assessment that they did was you know, initiated due to a California legislator, actually, I think a senator in state senator in Fremont, I'm forgetting his name, kind of bringing this forward as, you know, something that, you know, him and I assume other legislators wanted the scientists of California to investigate. And so that's kind of where it started. And then I know that there was a bill at some point, on the floor to propose, I think, putting like warning labels on foods that contain the dyes, but I don't know what's happened with that, and I haven't heard anything about it. So it kind of becomes just the, comes down to like how quickly our bureaucracy can move on these questions and issues. So I don't really know. It would be great if the FDA, then did another review of the evidence based on this stuff, because they definitely follow California scientists pretty closely, given the great scientists we have, and the questions we're trying to ask and answer. So sorry, that's the best I can do, but it's a really good question, and I do hope to see more come out in the near future.
Yes, thank you for being as much of a policy expert as any of us could be in this forum. We know you are not all on that side of it, so we appreciate your insight.
Yeah, of course.
The next question is asking, is it known how food dyes are metabolized by the body? Are they mostly excreted in the urine or accumulate in specific tissues?
Yeah, that's a good question. I'm not a toxicologist, so I actually don't know, but there's definitely a lot of information on that in the report. It's a long report, but it has a table of contents that can direct you to where there's discussion of this. But this is definitely something the toxicologists on the team looked into, and I think there is a good body of literature on what happens to them. So I'm not sure, but great question.
Great. And then the final question that we have for now, though anyone who has last minute questions, please feel free to submit them, is for the randomized trials that linked food dye with behavioral issues. Can there be some placebo effects, since dyed versus non-dyed foods are kind of hard not to be able to differentiate, or a reverse relationship like children with ADD having a preference for colorful foods?
Yeah, so this is a really good question. This gets to the heart of what it means to design a good randomized controlled trial to answer a question like this, because there are observational studies, but they're all obviously flawed, can be very flawed due to these kinds of issues. So I know the two big trials that I mentioned. One of them I know better than the other. It's this study done in Southampton in the UK, and that was a double-blinded study. So I believe they blinded by basically having the kids consume the dye in like some beverage, where maybe they couldn't see the color of the beverage or something like that, or they created some color mimicking kind of compound that wasn't quite the dyes to give them a really similar appearance. So they definitely tried to kind of reduce the bias as much as possible in that way. And I think usually there are tests for the effectiveness of a placebo in disguising the treatment assignment, that can take the form of asking the child or the family, like, do they think that they're in the treatment or the placebo group? And if the rates are similar, then you think that, okay, then we did a pretty good job. And you'd be surprised, a lot of people think that they're in the treatment group. It's interesting. But and then there are other steps to kind of so for the reverse relationship, potential for a reverse relationship, because it's a randomized trial, and the consumption of the AFCs is being assigned to the kids, they ideally their preferences are taken away from that, assuming they stay enrolled in the trial and everything. So that's addressed in a randomized trial setting, but not in an observational setting. Definitely in observational studies, you might have kids who have those symptoms tending to have a preference for colorful foods, for sure. And then there are other steps that are taken to try to make the reduce bias, like usually to assess attention and behavior. They have multiple raters, like include the parents, but also like external raters like the teacher and just research staff. So and all those people are ideally also blinded to the treatment assignment, whether in the treatment or control arm. So yeah, so that's they definitely try to address all of those issues for sure. But yeah, it's no study is perfect, definitely, if I've learned anything in epidemiology. Yeah.
Especially when it's all by or a lot by whatchamacallit, user reporting.
Yes. Yeah, definitely self-reporting. Exactly. That's a huge bias. So we love having objective biomarkers, like something you can measure in the blood, but obviously here the focus is on things that have to be observed. Yeah. So yeah, great question.
Great. Well, thank you so much to both of our speakers. We'll give one last round of applause. We really appreciate you guys coming out here tonight and speaking with us. And if anyone is interested in more topics, we've got a bunch of resources from these two speakers. So that should be great. And I'll give it to Oksana to take us out.
Yeah, thank you again to Naren and Ruwan for their wonderful talks tonight. If you enjoyed these presentations and are not currently set up to hear about future talks, you can join our listserv using the QR code on the screen or connect with us via the links sent through the chat. We will also upload a recording of the seminar to our YouTube page where you can find our archives and send out a post-seminar email with resources from our speakers so that you can follow up on these topics if you are so interested. We will be back next month on May 16, both in person at the Central Library for the first time in 3 years, and also on Zoom. Until then, we'd like to thank our speakers for their great talks one last time, and also thank you, our audience, for joining us today.
Thank you, everyone. Thank you, speakers. Thank you, organizers. Thank you, audience. Have a good night.